Hi, everybody. So this is uh, chapter 9, um, genetically modified organisms. Um, and we are going to be talking about gene expressions. So how we go from a genotype to a phenotype, mutations, stem cells, and cloning. So it's a big one. And there are a lot of learning outcomes for this one. Some of them are a little more straightforward than others, a little more complex. So this one involves quite a few things, whereas just this is pretty one sentence answer. All right, so let's talk about protein synthesis and gene expression. So remember, we have a genome, and a gene codes for one protein. And I did draw this on a previous slide. The genes are going to be on the chromosomes in the nucleus of a eukaryotic cell. And in order to turn that into a protein, we need to copy it into a message. This is called messenger RNA. And then that is going to leave the nucleus, and once it's out in the cytoplasm, then that messenger RNA is going to be translated into protein. So remember, DNA is double-stranded, and messenger RNA is going to be single-stranded. And this is the process that you need to know. In fact, what would be great is if you could redraw this process and explain what's happening at each step. So you've got the DNA, and on the DNA you have your gene. And what happens is the strands get separated, and instead of copying another copy of DNA like we would do in DNA replication, there is a polymerase that copies onto the RNA. So if you look here, the RNA is a match to this top strand of DNA. So this is how the message gets rewritten or transcribed, right? If you were to rewrite your notes, you would be transcribing them. And so this process is called transcription. And remember, RNA is different from DNA in that it uses uracil, and it's got a different sugar, and it's single-stranded. So now that messenger RNA, and this is the process we need to know, that messenger RNA is going to go out into the cytoplasm, where it's going to meet up with a ribosome, and the ribosome, along with other types of RNA, so there's ribosomal RNA, there's messenger RNA, and there's transfer RNA. Those together are going to act like enzymes to translate the RNA into protein. So if these are nucleic acids, right, they're the same macromolecule. Protein is a different macromolecule. So you're rewriting it, same language, you're translating it into a different language. So here's the transcription. We open up the DNA, and a polymerase copies on that complementary messenger RNA. And then the messenger RNA is going to go out into the cytoplasm, where it's going to meet up with this thing called a ribosome, and it's also made of RNA. And it's got two parts, and it chomps down onto the messenger RNA. The messenger RNA fits in here. And then it houses these things called tRNA. And tRNA have, they have a little bit of RNA that complements the messenger RNA. So they fit like little puzzle pieces, and they bring along on the other end of them an amino acid. And that amino acid is the one that these three little nucleotides code for. Three nucleotides together that code for an amino acid are called a codon. And, the anticodon, that is the matching one. And so what happens is inside the, and I don't know if I have I don't have a slide that shows it. It probably is in extreme detail in the book, and I just pulled it out because I didn't want to overwhelm you with the extreme detail. But what happens is, you've got your messenger RNA and you've got your ribosome. And the ribosome has these two open sites. They're not really catalytic sites because it's not really an enzyme, but they're places where the tRNA comes in. And the tRNA comes in and it matches, its anticodon matches to that specific spot on the messenger RNA, and it brings along its amino acid. And then the messenger or the ribosome moves along in this direction, and the tRNA stays there, and so it ends up. I wish I could erase this, but I don't know how to do that because I'm not that fancy. It ends up over here. Maybe I'll change the color. Let's make it. So this tRNA moves or stays in place, and the ribosome moves. And now a new codon is exposed. So a new transfer RNA comes in and it brings its amino acid. And now you've got two amino acids together and they can form a peptide bond. And so then this tRNA leaves and this one stays in place and the ribosome moves again. And this one ends up over here and a new tRNA comes in and it brings a new amino acid, and you end up with this growing amino acid chain. And eventually it reaches what we call a stop codon, and the whole thing falls apart and it releases your polypeptide ready to be folded into a protein. So proteins put together one amino acid at a time, and eventually that full protein is released. So remember that codon is 3 nucleotides. There are 64 combinations, but only 61 of them code for an amino acid. Of course, we have to have a stop codon. We have to have a place to say, nope, that's the end of the message. It's like a period. And what's fun is, so we all have the same DNA possibilities. We also all follow the same code. So for example, these particular codons all code for an amino acid called threonine. In humans, dogs, birds, lizards, bacteria, fungi, starfish, etc. And it's redundant in the sense that it's not one codon always for, so that's universal, but not one codon where it all, starfish, dogs, et cetera, universal. Now back up here, it's redundant. This thing is called the wobble effect because you can have multiple codons that all code for one amino acid. And what is important about this, and let's see if this table helps. Yeah. So it's not always perfect that 4 will code for here, over here. You can have 2 that will code for 1, over here it's 2. But what's important about the fact that this is redundant, it's called the wobble effect. It's kind of fun, wobble, is that if you have a mutation in the DNA or a mistake in the messenger RNA, it doesn't always end up meaning that the amino acid is going to be different or wrong. It doesn't always mean that you're going to end up with the wrong protein. So as long as you have a UC in this particular codon, it doesn't matter what the last nucleotide is. You're going to get the amino acid serine. And so that's important for us. Oh yeah, and a video within a video, we're not going to do that. So mutations. I just said it is very possible that you can have a mutation and it's not going to make a difference. So it's going to have no change. It is possible that that particular amino acid was very important and it did change it, and you end up with a non-functional protein. Like one way of doing it is you accidentally introduce a stop codon. Like here, if you have a different third amino acid and you meant cysteine and you end up with a stop codon, that puts a period in the middle of a sentence. and that's a problem, right? Then we don't get a functional protein. You might get a slightly different protein, one that folds differently, because remember, it's those variable groups, those side chains of the amino acids that bond together or interact together, or they're hydrophobic or hydrophilic and they want to tuck in or out of the protein. And if you change it for one that is wildly different at a critical part of that protein, then you can end up with a different protein. Or you can end up with a non-functional protein that way too. So yeah, here, this is called a substitution mutation, where you change just one nucleotide and you end up with a different amino acid. So you substituted a different amino acid. A silent mutation is the one where it doesn't do anything, thanks to the wobble effect, or something like that. It just really doesn't matter. Oh, yeah, there's the wobble effect. Frameshift mutations are a different thing altogether. They're not a substitution mutation. They're an addition or a deletion. And so if you've got a DNA sequence, which makes an RNA sequence, and you stick a random nucleotide in there, it's like shoving a letter in the middle of your sentence, and all the rest of the letters in all the words, either shove down the line, or if you delete one, they move up in the line. So I have an example here that I wrote for you. So if you have the fat cat ate the red rat, that's your your sentence. Those are your codons. But if you stick one in there, then everything else moves down. So this T moves, this A, this T, this E, etc., all the way down. And now we end up with like not words. So frameshift mutations are extremely damaging. So this would be an insertion frameshift. This would be a deletion. If we took the T out, then we have, instead of cat, we have like ka and tet and her and edrat. And so, yeah, that's definitely, it's a non-functional. So frameshift mutations are the most damaging. Okay, so gene expression. So remember, we have 2 copies of every allele. And so on our DNA, I don't know if you remember, I had that picture where the DNA is, wrapped around proteins to make a nucleosome. And so there are lots of ways that we can regulate whether or not a gene gets expressed. We can do it by, regulating how it's wrapped and when it comes unwrapped. You can regulate it right at the transcription level. By starting or stopping the polymerase, by physically blocking the polymerase. Let's see, do I have a picture of that? Yeah, I do. So you can do it by physically blocking the polymerase, or starting up the polymerase by removing a block. You can do it by unwrapping it from those proteins. You can do it by chewing up the mRNA or rearranging the mRNA at that level. And then you can also do it by regulating the ribosome at the point of translation. But this is the one that the textbook, it really kind of wants to illustrate. So there's 2 different ways that you can regulate gene expression at the transcription label. So transcription is the one where you're transcribing, you're rewriting. So this is the one where we're making the messenger RNA from the DNA. And there are 2 ways that you can do this. You can have a protein that's a repressor that binds onto the DNA, and it prevents the polymerase from even getting in there to do its job. And then you can regulate whether or not that repressor can bind. So you can change that repressor protein and make it fall off, or you can change that repressor protein and put it back on. You can also have activator proteins, which are going to say, hey, Polymerase, like this particular gene needs to be transcribed. So it's kind of like an advertising sign to have that activator protein there. It attracts the polymerase. And then, like I said, there are other ways of doing it. Once you've made the mRNA, you can cut it up. You can also cut up the protein after you've made it too. So we can use these kind of tools to our advantage for things like, well, we actually we call this cloning in microbiology, where we take a gene and we have transcribed it from the DNA of one microbe, and we put it in a little circle of DNA, and we put that little circle of DNA in a bacterium, and now the bacteria work as like little factories to continually make that protein that that gene coded for. And in this case, they're talking about bovine growth hormone, and putting that bovine growth hormone into things. Oh yeah, so you can take the gene and you can cut out the gene, and then you put it in a little circle of DNA, and you put that little circle of DNA into a bacterium, and then the bacteria work to keep making that for you, and they can keep making that hormone and keep making that hormone and keep making that hormone. And then you can take that big vat of bacteria and you can spin it all down, and you can extract that protein, and then you can have pure recombinant, that's what the little R is for, recombinant bovine growth hormone. And then you can take that and you can inject that back into cows and give them a whopping dose of hormones. And the reason why people do this is because it increases milk production. And there's some trade-off to that, but you know, as with pretty much everything, it's money. And so if we can house one cow and get that much more milk, then you know they're going to do it. People say that there's that there's no detectable difference, but drinking milk that comes from a cow that's meant for baby cows, not humans, that is now injected with growth hormones that are meant to be for cows, not humans. There is something to be said about this, and there's definitely a lot of research. And I am not against genetically modified organisms. I'm not making a blanket statement about GMOs, but this one's a little interesting and worth investigating. So let's talk about genetically modified organisms. Like, since we've been cultivating crops, since we have went from hunters and gatherers to sedentary stationary farmers, we started to cross plants and only save the seeds from the plants that gave us the best yields and the the tastiest food, and et cetera, et cetera. And so we have been genetically modifying organisms for eons. But we've been doing it slowly. What people have a hard time with is when you kind of do it quickly, and when you do it maybe without the benefit of natural selection, where you put something in and the environment doesn't tell us that there is a second effect. Yeah, so if it's a living organism, we're going to call it transgenic organism. There are certainly some reasons why we might want to modify particular organisms. I can say when I was, it's down here. When I was at Michigan State looking for what kind of graduate program I wanted to get into for microbiology, one of the professors, he was instrumental in developing golden rice. So golden rice doesn't, or rice in general, non-genetically modified rice doesn't have a lot of like nutritional value. It's just starchy. And if you could take beta carotene, which is an antioxidant, a vitamin, and you can cause the rice kernels to express that gene. So the rice makes beta carotene and turns yellow. Then when you ingest that rice, you get the benefit of beta carotene as well. And so, people who maybe don't have access to foods that carry beta carotene is in carrots and tomatoes, and it's that red orange color, and it's good for your eyes. It's an antioxidant. So it's a very beneficial thing to put into rice, which wouldn't otherwise have it. And people who don't have access to alternatives, this genetically modified organism can improve the health of those populations. Some people do have some concerns though. Again, if if you're only selecting, like, let's say you put nicotine in something because you are trying to prevent insects from eating it. Well, then, how much of that nicotine is expressed in the corn kernel or the potato underground? I'm just throwing stuff out there, but it might be possible. And then if you think on an ecosystem level, what happens when microbes and insects and other organisms in that ecosystem didn't co-evolve with the change that you made? How are they going to respond, and what are the downstream effects of making this change? Yeah, okay, here you go. What are the downstream effects? So if you mess up or mess with, not mess up, but if you mess with salmon, are you disrupting the food web? So there's another one. And then when it comes to using animals to create products for humans, so you can clone into animals to produce something, say like something in the milk, blood clotting proteins in the milk, and then harvest that milk from the goat. There are like some huge ethical concerns about that, because is that healthy for the goat? Is that a good lifestyle for the goat? There's definitely some concerns there. Genetically modified humans, people really tend to stand on one side or the other about things called stem cells. Stem cells are naive cells, so they've been freshly made, either in the umbilical cord or the bone marrow, and they haven't decided what their job in the body is going to be. So they don't know if they're going to be a bone cell, a liver cell, a skin cell. They don't know if they're going to specialize and become gametes. They are completely blank slates, which is why they're really good for using for research. They're also possibly good for treating diseases. So if you happen to have a genetic disease, or you have some tissue or organ degrading disease, or even a neurological disease, these cells might be able to replace or supplement the ones that are damaged or wrong in some way, not functioning correctly. And so there certainly can be some very promising uses for stem cells besides just research, but in direct application. And the way that works is you take that naive cell and you program it to be a healthy cell that you want to fix. And so the way we program it is, remember, viruses have to use a Oh, you haven't gotten to that chapter yet. I just wrote it, but you're not remembering. So viruses have to have a host cell in order to replicate. And what we can do is we can take a virus and we can take out all their genetic material, all their program, and we can use them as a vessel to put the gene in that we want. And then when they infect, and I'm doing big air quotes because it's not really an infection, but when they go inside of the host cell like they would do if they were to cause an infection, they're incapable of causing an infection because the DNA or RNA that they carry is what we put in there. And so they just disappear, but the genetic material stays, and the genetic material gets copied, it gets taken up by that cell and copied and used to replace or fix the defective proteins in that cell. That's really cool. So, these are pretty big, hot topics, but they're not necessarily easily blanketed as good or bad. So hopefully you can go through these learning outcomes and speak to each one of those. And I am going to stop the recording here.